Welcome to UMDF's Ask the Mito Doc monthly webcast. I'm Kara Strittmatter, UMDF Director of Education and Support Services, and tonight we will hear from Dr. Austin Larson from Children's Hospital of Colorado, Dr. Eva Morava Kosic, and Dr. Ibrahim El Sharkoy, both from Icahn School of Medicine at Mount Sinai, New York, New York. Our Ask the Mito Doc webcasts are made possible through the generosity of our sponsors. So we ask that you join us in thanking the Edith Trees Charitable Trust and UCB. Next week, we join many of our patient advocacy groups around the world for Mitochondrial Disease Awareness Week. We hope you will join us in raising awareness on social media and in your own backyards. UMDF has many ideas and tools for you to get the word out about Mito, so visit umdf.org today. One great way is to participate in an event in your area. UMDF has at least 15 walks happening this fall, so please visit energyforlifewalk.org to find a walk near you, or walk virtually. Fall is a beautiful time to get out with your loved ones. Thanks to our friends at UCB, our no-cost genetic testing program continues to grow for our patients and family. Please scan the code on your screen or visit umdf.org. The link is front and center on our page. If you or a loved one has been diagnosed with MELAS, our friends at Tacento Therapeutics would love to hear your patient journey to help shape an upcoming clinical trial. Visit umdf.org/studies and surveys for more information. On the heels of a fabulous conference in Cleveland, we are already making plans for St. Louis for Mitochondrial Medicine 2025, June 18th through the 21st. Save the dates now, and more details will come at umdfconference.org. Please note, the information provided in this webcast is for general information only and should not be relied upon as a substitute for professional consultation with a qualified healthcare provider familiar with your individual medical needs. Please consult your primary physician and/or specialist before changing the course of your care. So let's get started. I will ask our first speaker to join us and share slides to help us all better understand genetic testing.
Good evening. Thank you for having me on this topic. I'm Austin Larson. I'm a medical geneticist. I see most of the patients with mitochondrial disease that I care for with my colleagues at Children's Hospital Colorado, and I'm on the faculty at University of Colorado. I think it's always important when you're approaching any topic to start with the why. So the why for genetic testing is that in some cases, we may identify a specific mitochondrial disease that has a treatment that we wouldn't know to implement in the absence of a genetic diagnosis. The genetic diagnosis gives us an etiology or a mechanism. You'll probably hear me say those words multiple times this evening: etiologic diagnosis or mechanistic diagnosis. It's really important both for existing treatments, but also for the potential to participate in a clinical trial. So increasingly, clinical trials in the area of mitochondrial disease are limited to patients with specific genetic diagnoses. And the reason for that is so that we can make sure that there's the optimal risk-benefit ratio, that we're actually giving a potential treatment that has the potential to work on the specific mechanism of disease that the subjects have. Even if there's not a specific clinical trial, gathering people together with the same genetic diagnosis can be really important to lay the groundwork for future clinical trials. We call that natural history or clinical trial readiness. So accurately finding etiologic or mechanistic or genetic diagnoses is really critical to lay that groundwork for future treatments. Another reason that genetic testing is really important when we're evaluating patients who may have mitochondrial disease is that many patients actually have alternative diagnoses. So a lot of the symptoms of mitochondrial disease may not be specific to just mitochondrial mechanisms. There may be other mechanisms of disease that can look very similar clinically. And it's important to identify those diagnoses, again, to be able to accurately establish a mechanism of the symptoms and to be able to provide specific treatments. Sometimes we're in the opposite scenario. So sometimes I'm not talking to an audience about mitochondrial disease. I might be talking to an ophthalmology audience or an endocrinology audience. And a lot of times that the outcome of facilitating or helping those providers to do more genetic testing is that we actually find mitochondrial disease in unlikely places or places where people didn't know that they needed to be looking for mitochondrial disease. So establishing a precedent of using broad genetic testing and using it earlier in a diagnostic evaluation can lead to identifying more patients with mitochondrial disease. Lastly, particularly in the field of mitochondrial disease, even more so than other areas of genetics and metabolism, there's the potential for recurrence within a family and having multiple affected family members. So finding a genetic diagnosis can be really critical to understand who else in the family may need evaluation for mitochondrial disease. So there's one big division in the area of mitochondrial diagnostics, which is the idea of mitochondrial DNA versus nuclear DNA. So for these first 20 minutes of the session, I'm going to focus on nuclear DNA. And Dr. El-Sharkawi is going to focus on understanding the significance of mitochondrial DNA testing results. So when we talk about the approach to nuclear DNA testing, we're usually talking about one of three things: either whole genome sequencing, whole exome sequencing, or targeted sequencing. And whole genome sequencing is kind of the most recent of these tests to be developed and put into broad clinical practice. It is increasingly available, although there are still some barriers in terms of either provider comfort with newer tests or insurance coverage. But increasingly, whole genome sequencing is available. It is the most expensive of the genetic testing modalities, but it has the broadest coverage and it has the ability to identify some types of genetic variation that aren't as well identified by exome sequencing or targeted sequencing. Exome sequencing is kind of in a middle ground in terms of cost. It covers the coding regions of almost all genes. So that's roughly 20,000 genes. The coding regions of the genes means the areas of the genome that are actually responsible for making proteins. The other 99% of the genome is either structural or regulatory or completely inert and insignificant. But that exome gives us the highest concentration of being able to assess the high yield areas of the genome. Lastly, if we have a really strong clinical intuition about the basis for a patient's symptoms, we may be able to use targeted sequencing or panels. That would be the least expensive option. It also gives us the deepest coverage for the genes of interest. But increasingly, as you're working with providers who are trying to arrive at an accurate genetic diagnosis, more and more people are going to be using whole exome and whole genome sequencing as a first-line test. So I'm going to walk you through the rationale for why we may be moving towards using broader testing. 10 or 15 years ago, I started in the field of medical genetics 10 years ago, and I really remember the process of changing our strategies as genetic technologies have improved. So when I first started in the field, there was a lot of very, very detailed work went into examining a patient, looking through records, looking through radiology results and lab tests, and compiling a oftentimes 20-page long clinical note with the goal of that clinical evaluation to try to come up with an idea for a single gene or a single genetic diagnosis, and then to pick out the one right test out of the hundreds or thousands of different tests that might be able to diagnose a patient. So while there's a lot of kind of romanticism about an earlier era of clinical diagnostics and diagnosticians sort of thinking very carefully, doing what we call phenotyping, and then arriving at a genetic diagnosis and just testing for that one possible diagnosis, the reality was that we just didn't find diagnoses very often when we were using that process. It's incredibly hard, and honestly just beyond the confines of human cognition to be able to accurately pick out a clinical diagnosis when there are thousands of possible different genetic diagnoses. Some of the other examples of tests that I'm showing you here are looking for different types of genetic mechanisms, looking at the mitochondrial DNA specifically, maybe doing biochemical testing. But all of these tests kind of have the same principle, which is that you have to have a very specific clinical scenario to be able to pick the right test. Contrasting that with the current era, whole genome sequencing or whole exome sequencing for the most part really encompasses almost all of those thousands of different tests that we were doing 10 years ago. Whole genome sequencing has very good sensitivity when you compare it to doing metabolic testing, even for patients with metabolic disorders. Whole genome sequencing has really good sensitivity for picking up deletions or differences in the copy number of DNA. As of the last few years, whole genome sequencing has really good techniques for assessing the mitochondrial DNA. So Dr. El-Sharkawi will tell you more about mitochondrial DNA, but increasingly, even that is encompassed within this one single, very broad test. So if I'm asked to conduct a diagnostic evaluation and try to diagnose a patient, my bias, or if I'm going to make an error, I would like to err on the side of doing a broader test because the errors are asymmetrical. So in one direction, maybe I did a broad test where occasionally I could have done a narrower test and identified that patient slightly, with a slightly different strategy, but that's dramatically outweighed by the times when I'm honestly just surprised and humbled by the diagnoses that come out of having that hypothesis-free, broad, unbiased approach to considering all of the possible genetic diagnoses. So this is a picture of a psychologist named Thomas Gilovich who studies decision-making. And his quote really, I think, encompasses or encapsulates the practice of clinical medicine and particularly diagnostics really nicely. So his quote is, "The world does not play fair. Instead of providing us with clear information that would enable us to know better, it presents us with messy data that are random, incomplete, unrepresentative, ambiguous, inconsistent, unpalatable, or secondhand." The point being that it's very hard to arrive at a clinical diagnosis in the absence of that kind of broad and hypothesis-free, unbiased testing. The other really big advantage of doing a broad test, in addition to the fact that we're more likely to identify a diagnosis, is the fact that if that test is negative, there's much more value in that negative test than if we had done a narrower test that was less sensitive. So I'm using some kind of technical terms here, but sensitivity, the technical meaning of sensitivity is the likelihood that a test will detect the presence of a diagnosis. Negative predictive value, or NPV, is the likelihood that a negative test result is a true reflection of the absence of a diagnosis instead of just a failure to detect it. So what this means practically is that exome and genome sequencing, if those results are negative, it's not 100%. It's never 100% certain that that means that a genetic diagnosis is not present. But the better these tests get, the more sensitive they get, the more value there is in that negative result saying, okay, we've done a really good test to consider the possibility of a genetic diagnosis. And now we need to think about other possibilities, because as we get to exome and genome sequencing, there actually really is significant negative predictive value. Again, it's not 100%, but it's not zero either in terms of how we use that information going forward. So I hear from a lot of folks who I see in clinic, it took me so long to get in to see you. This is so frustrating that there aren't more geneticists around. And I agree. I really wish there were more geneticists. I run a training program. I train geneticists. I think that would be good for medicine if there were more people in the specialty. But for the time being, and at least for the foreseeable future, genetic expertise will be a limited resource. So question is, when is it the most valuable? And as we move from this paradigm of having really narrow and targeted tests that require very specific clinical scenarios to be useful, and we move more towards a broad test that broadly encompasses all of the possible genetic diagnoses, or almost all of them, the allocation of that expertise can change. So I think there's more value in having genetics expertise on the side of interpreting and applying those results in the clinical context. And there's less value of me choosing the test. For the most part, I know what test I'm going to choose if I'm doing a diagnostic evaluation. And I think it's appropriate for that testing to happen earlier in the diagnostic evaluation in some cases. So when you do a really broad test, you're going to get a lot of information out of that. So it's really important to remember that there's both an art and a science to genetic testing. So the science is generating the genetic data and what we call the wet lab process and some of the computer analytics. And then there's an art. At the end of the day, there are human beings, both in the lab and in the clinic, that are using their expertise and giving their best judgment calls about the clinical significance of all the rare genetic changes. And the outcome, which I hear a lot about from different patients and from other providers, is how come there's all these variants of uncertain significance? So I'm going to talk through that a little bit, and Dr. Morava is going to talk with us about how we have some new tools that may help us to resolve some of the variants of uncertain significance. So for exome or genome sequencing in the lab, they're taking a DNA sample. Often that's from a cheek swab now. Sometimes it's a blood sample. Sometimes it's a muscle biopsy. And they're generating sequence. All these tests that we're talking about are what's called short read sequencing tests. They kind of blanket the whole genome with little snippets of genetic information. And then they compare that to what's called the reference sequence. And they look at how common genetic variation is in the healthy population at any given position. And they apply that, some databases that we have. We have now a database called Nomad with over 800,000 people. And that's a really powerful data source to know what's common and what's uncommon. So those things that are pretty common in the population, for the most part, we don't need to worry about them. Those labs are going to use internal data to see what's common in their lab versus uncommon in their lab. They're going to use computer predictions, which are getting better and better. So there's some really smart data scientists who are using machine learning and AI to help us understand how genetic changes impact the protein products that are resulting from those genes that have variants within them. And then at the end of that process, there's a lab director, often someone with PhD training, who sits down and thinks about, given all the evidence that has been gathered, is this something that we should put on a clinical report? And if we put it on a clinical report, should we tell the clinician that this is likely pathogenic, that it's likely benign, or that it's somewhere in the middle, which is called a variant of uncertain significance? And there's accepted criteria for how you classify the variants into those categories. So for myself, as a medical genetics provider in the clinic, I get that report from a lab with maybe a diagnostic result, maybe an uncertain result. And my job is to talk with the patient about what I think the clinical significance of the genetic testing results are and what the next steps are. Is there a follow-up test, like what Dr. Marava is going to talk about, that can help us to resolve the uncertainty around a particular variant? Or maybe I feel like the clinical diagnosis, now that I have a genetic result to consider, a diagnosis where I'm not just thinking about thousands of possible diagnoses, but my clinical job may be to think about one diagnosis and just say yes or no. Sometimes the clinical features can look pretty different when you have that lens. And I may be able to say to someone, even though this is a variant of uncertain significance, the clinical features here, maybe the imaging or the biochemistry or exam findings or family history, maybe those things are so specific that I can tell you right now that this is a genetic diagnosis. And if I can say that, how certain am I about that? Am I certain enough to prescribe a medication? Am I certain enough to do something like organ transplantation or some very high risk type of intervention? Those are the judgment calls that I make about my certainty of the significance of a genetic result. Is it appropriate to test other family members for those results? So that's the thought process that I'm going through. So I hope that you all had a chance to learn not just what exome and genome sequencing are, but sort of the thought process behind why we might use those tests, and also a little bit of not just the science behind it, but the art of getting those results and applying them to patient care. So I'm going to turn it over now to Dr. El-Sharkawi to talk about mitochondrial DNA testing.
I'm just going to share my screen here. Okay, assuming everyone can see my screen okay. Hi, everyone. I'm Ibrahim El-Sharkawi. I also go by Ibi. I'm an attending and faculty at Mount Sinai Department of Genetics. I work closely with Dr. Marova. All right, so without further ado, so I'll be talking about mitochondrial DNA and how we approach mitochondrial variant, a mitochondrial variant of interest. I'll use a real life case to exemplify how we think through these variants when they come up. But before we do that, before we dive into the case, just a couple of intro remarks in terms to make sure we're all on the same page. So mitochondrial DNA variants, we used to call them mutations, but now we call them variants. Any changes in the DNA from what we understand to be the reference range of what these, or the reference genome, can show heteroplasmy. What heteroplasmy means is different levels, different percentages of the degree of mutation, the mutation load, if you will. So for example, if I say there's 50% heteroplasmy in the tissue we looked at, be that blood, cheek swab, or urine, that means in that sample, 50% of the mitochondrial DNA that we looked at had the variant, the mutation, and 50% do not. 50% are what we call wild type. And these percentages can differ in different tissues as well. So if you look at urine, it might be a little higher than in blood, and so on and so forth. And heteroplasmy can also be different among siblings with the same variant and from mom to children as well. And we'll explain why that is in a little bit. And so we all have variants in our DNA. Variants don't always mean disease. They don't always disrupt how the gene works. And so they're not always disease causing. So we all have variants in our DNA. That's just a normal part of life, of evolution. Most of these variants don't mean anything. They don't change how the gene works, and so they're considered benign. And some are disease causing, known as pathogenic. And then often, because we're still in the early stages of really understanding the genome and all the complexities of genetics, we're still we can identify a new variant quite well, reasonably well. Our tests can detect a variant, a change, but we're not always sure of how relevant this variant is. Is it disease causing? Is it a pathogenic variant? Or is it one of those benign variants, but we just don't know enough about it to call it a benign variant? And so, as Dr. Larson said, this is a variant of unknown significance, and this can happen with the nuclear DNA or with the mitochondrial DNA. So just to carry on where Dr. Larson left us, we talked about nuclear DNA. This is the nucleus where our quote unquote main DNA is the nuclear DNA. This is where we have 2 copies of every gene. One is inherited from mom, one is from dad. And this is the rest of our cell, which makes up all our tissues and all our organs. And these are the mitochondria. We have many mitochondria in each cell. And inside the mitochondria, we have mitochondrial DNA. So mitochondria have their own separate DNA that is passed down from mother to offspring. So this is what mitochondrial DNA looks like. And these are all the genes within the mitochondrial DNA. So if you see ND5, ND4, if you see the variant is somewhere along, is in one of these genes, it'll say variant and give you a number. And that number basically tells us the position we're in this circular piece of DNA here. And it'll tell us the involved gene. So a variant at ND4, for example, means there's complex 1. This gene is involved in complex 1 activity, and a variant in MT-ATPase6, for example, would be a variant in the gene that helps control complex 5. And so why is heteroplasmy different? Why do we have why can we have a mom with 80% heteroplasmy in blood and a child with 40% heteroplasmy, for example? And I'll explain why using this picture here. Like as they say, a picture speaks 1,000 words. And it's worth noting, of course, that 100% heteroplasmy, quote unquote, can occur, and then we call that homoplasmy. When a variant is seen in all the mitochondrial DNA we're looking at, we call that homoplasmy. And so the way heteroplasmy works is picture this. So you have, pretend these marbles here are all mitochondrial DNA. And then your yellow ones, your yellow circles here, are copies of mitochondrial DNA that have a specific disease-causing variant, a specific pathogenic variant. As our egg cells are forming, because of the way our cells randomly divide and randomly collect mitochondrial DNA and then divide and expand from there, just by luck of the draw, some of the egg cells might have more healthy mitochondria, and some may have more of the mitochondria with the mutation. And then, as you can see here, as you expand more and more and more, it can end up being very different from cell to cell, from organ to organ. And this is just another way of looking at it, a different way of understanding wild type, so quote unquote normal mitochondrial DNA and mutant DNA. And then generally speaking, and of course, this isn't a hard and fast rule, but generally speaking, if you have a high heteroplasmy level, a high mutation load, you'll have a severe phenotype, what we call severe disease. If you have about 50% heteroplasmy or so, it's a milder phenotype. And then usually if you're at 10, 15, 5% heteroplasmy, you may be asymptomatic or have very mild disease. So let's run through this case. So we have a new patient with suspected Mito, 6-year-old boy, has new seizures, previously well, suddenly developed new seizures and an elevated lactic acid. A brain MRI shows us findings consistent with Leigh syndrome. metabolic testing, which you'll also hear us refer to that as a small molecule workup, as biochemical testing. This is where we're looking at amino acids, urine organic acids, anything to help give us a clue or give us or support the diagnosis is overall unremarkable. And remember, you can have tried and true mitochondrial disease and still have completely normal biochemical testing. But it certainly helps. It can offer a good clue if it's positive. But being negative doesn't rule out anything. And so appropriately, we sent genetic testing. We sent whole exome sequencing with mitochondrial DNA, which hopefully we all understand a bit better now following Dr. Larson's talk. And we found 2 variants of interest. We found an MTND3 variant at 46% heteroplasmy in blood and an MTTR variant at 50% heteroplasmy in blood. So 2 potential causes, 2 potential etiologies here. And both variants are classified as variants of unknown significance. So pause. This is why can we still not say this is true confirmed mitochondrial disease? Certainly the brain MRI shows Lee syndrome. Certainly the presentation is very consistent with mitochondrial disease. And now look at this. We have 2 variants in mitochondrial DNA at 50% heteroplasmy. How can we... But despite all this, despite the very, very convincing data, and even though I'm convinced by this and you may be convinced by this, we cannot say definitively that this is mitochondrial, primary mitochondrial disease, because these variants may still be variants of unknown. They are variants of unknown significance, and they may in fact be benign variants. So what do we do about it? How do we better understand this? Because establishing a true diagnosis is important for all the reasons Dr. Larson mentioned, and establishing a true molecular diagnosis might open up avenues for clinical trials. So it's very important that we try, if we can, to arrive at a diagnosis. We're not always successful because of how complicated it can get, which hopefully the next few slides will illustrate. So what do we do next? Next, as medical geneticists, we put our thinking caps on, we look at the variant, we look at online databases, we look at where this variant has been reported before, if it has been reported before, and we think through carefully this variant and whether or not it might disrupt the gene function and cause disease. The other thing we do, the next thing we do, is we can test mom. So mom, who's completely asymptomatic, ended up having the ND3 variant at 90% heteroplasmy in a cheek swab, in a buccal swab, and the MTTR variant at 3% heteroplasmy. So this makes us a little more suspicious for the MTTR variant and less suspicious for the MTND3 variant, because mom has a high mutation load, a high 90% heteroplasmy in a cheek swab, and this is what a buccal swab looks like here. We swab the inside of your cheek and we can send it on to the lab, and we can avoid a blood draw that way. And mom's completely asymptomatic. We'd expect that if mom really did, if this was a true pathogenic variant at such high levels in mom, she might have some of the symptoms as well. So that's one way we can investigate a variant further. And then there are other things we could do in a research avenue, things like mitochondrial cybrids and so on. It can get a little more complicated. But next clinical steps can include a muscle biopsy. So back in the day, maybe before I even started my general pediatrics training, muscle biopsies we would often get muscle biopsies in patients in whom we suspected a mitochondrial disease before genetic testing got as good as it has been. Now, these days, in this day and age, we only get a muscle biopsy, which is an invasive procedure, only if we need it. If we need to get clarity on the diagnosis, so if we have a variant of unknown significance, but we still have high clinical suspicion for mitochondrial disease. So in this case, yes, we would get a muscle biopsy, and we looked at the muscle biopsy carefully, and Dr. Moravo will tell you exactly what we look at with all these newer clinical studies we can do to help us understand a diagnosis or arrive at a diagnosis. And here we identified both the variants that were around 50% heteroplasmy in this child's blood at 90% heteroplasmy in blood. And we look at complex activities, what we call ETC analysis. We looked at complex 1, complex 2, complex, so on. And we found that all complex activities were normal in this patient. And that again, does not rule anything out, but might make us a little less suspicious for the MTND3 variant. Because if you recall from the picture of the circular mitochondrial DNA, MTND3 is in complex 1. It helps, it's one subunit of complex 1, which is made up of, complex 1 is the biggest complex, and it's made up of many different subunits, ND3 being one of them. At 90% heteroplasmy, you might expect that complex 1 activity would take a bit of a hit with one of its subunits, if one of its subunits was so disrupted. But again, this doesn't really rule anything out. It's just something for us to ponder and think about. But hopefully this will show you how complicated it can get and why it doesn't prove anything definitively one way or the other. And then the other thing we did in this case was we looked under the microscope at what the cells look like, what the mitochondria look like, and we did indeed see that it is consistent with a mitochondrial disease myopathy in the muscle biopsy. So this is what a muscle biopsy kind of looks like under the microscope. And then if you have mutations or disease-causing variants in your mitochondrial DNA, you'll have disrupted mitochondria, and that over time changes the muscle cell. And this is one of many things we look at in a muscle biopsy. So we can look for the variant and heteroplasmy in other tissues. We can test blood, buccal, urine, buccal being the cheek swab, but we're not looking at your heart, we're not looking at your eyes, we're not looking at your brain, simply because we can't. It's so invasive to go in and look for heteroplasmy levels in these different tissues, and may not be all that helpful. And that just illustrates your blood and your urine and your buccal, your cheek swab, heteroplasmy levels are only telling us so much, but really what we need to be, but we're not looking at these internal organs which may have the bulk of the disease. So even with a true pathogenic variant, a 50% heteroplasmy may be a gray area, because will this be enough to cause symptoms? Will 50% heteroplasmy be enough in this particular gene and this particular patient to cause enough disruption to how the tissue and organ works or not? So for example, a mom with a classic MELAS M3243 variant at 30% heteroplasmy in blood may be more severely affected than her son with 60% heteroplasmy in blood in that same variant. So heteroplasmy level is a good general guide, but it's not always a hard and fast rule. It doesn't always give us the information we want it to tell us. So take-home messages here are interpreting mitochondrial DNA variants, even in true pathogenic variants, can be tricky because of all these reasons, heteroplasmy and how that's different in different tissues. On top of that, we have to think through variants of unknown significance. Often a muscle biopsy and some of these other tests may help give us further evidence for a diagnosis. And your mito doctor, your provider, will think through the variants and use the clinical or research tools we have to help us better understand the variants. And without further ado, that'll take you to Dr. Morava's talk, who will go over what these newer tests are. Thank you.
Thank you, Evie, and I'm honored to be here and talk to you about some novel diagnostic testing. This is heavy stuff, so hold on. I try to be slow and not overwhelming. First of all, I want to go back to this issue of mitochondrial complexes and how these mitochondrial complexes are important in providing us with energy. Ibi, when I'm sharing my screen, do you see floating participants, or is my slide clear?
Your slide's clear. We see your cursor, your mouse, but that's
Okay.
that's it.
Okay. All right. So you see here mitochondrial complexes 1 to 5. And if you recall, when we were talking about how most of your energy is produced, then these proteins, which are inside your mitochondrion, are really important to pump electrons and protons through the membrane, which is almost like making electricity, just instead in our little mitochondrial factories, we are producing energy. So one could use measuring the activity of these protein complexes as a diagnostic approach. And you heard Austin and Ibi talking about the genetic approach. And indeed, interestingly, especially compared to Europe, where muscle biopsies are still very common, and many centers start their diagnostics with taking a tissue sample and measuring these protein complexes one by one and measure whether their activity is comparable to that of a healthy individual. In the US, we are really trying to have a genetic diagnosis first. So we rarely ask our patients to undergo a muscle biopsy unless we found nothing by genetic testing, and then we are pretty much sure that the patient has a mitochondrial disease, just we cannot prove it by genetic ways. So if a biochemical test is indicated, so biochemical test when you measure mitochondrial enzyme complex or protein activities in a tissue biopsy, then you should go for a tissue which is really depending on mitochondrial energy production. And obviously, if you take skin, for example, the skin is not a highly energy dependent tissue. But if you take muscle, of course, your muscle is using a lot of energy. So if there is a mitochondrial disease there, then there is a good chance that you will be able to measure it. Kind of a weird idea, but sometimes we do that. If somebody is presenting with a mitochondrial disease and the heart is affected, sometimes a patient develops cardiomyopathy. There are cases when we take a sample, a biopsy from heart, because it's also a very high energy demanding tissue, and you can measure these proteins, these mitochondrial enzyme complexes in cardiac tissue as well. And of course, you can use fibroblasts as well. You can take a skin biopsy and then measure enzyme activities there. There are some labs offering enzyme assays or mitochondrial complex, protein complex measurements in blood. They are much less reliable. And unfortunately, skin biopsy, even though it's less invasive and would be a great alternative for the muscle biopsy, sometimes does not show the abnormalities. And it's not a technical error, but the biochemical changes are not always present in a skin sample. So that could be a good approach to do more diagnostics. But if it's negative, it doesn't rule out at all a potential mitochondrial disease. So muscle biopsy versus skin biopsy, just remember that not all mitofunction defects appear in fibroblasts. So I wanted to bring in a case for you of an adult patient with a very severe muscle weakness. We call that myopathy. And so here you see this young man, and if you just look at his posture, you see that his shoulder is sort of floppy, it's hanging, it is coming forward. You see this extra wrinkle here. And when I asked this young gentleman to raise his arms, he wouldn't be able to raise it above shoulder level. This we call a proximal myopathy. And usually this is not the first symptom a patient like that experiences. Most patients start with hip and girdle weakness, getting weak in their thigh muscles. And this gentleman had proximal weakness in the leg and in the upper limbs, and had also kidney failure. So because of this unique presentation, nothing else, just these two symptoms, we didn't have like an obvious diagnosis. After examining the patient, we are thinking about, could it be malus? Could it be NARP? Could it be Golgi? Based on the clinical features, we were not coming out with, oh, yes, I know this disease, I recognize, let's test this gene. So the mitochondrial specialists who were involved in this case, they ordered the genetic testing Austin was talking about, the whole exome sequencing, which is testing the coding regions of the nuclear DNA. So they were expecting that this patient would have most likely one or two pathogenic variants we called mutations in the past in a gene, but they couldn't predict which one. And so they just did this very broad test to hope to find something. But they didn't find anything. They just found two suspicious variants in a gene we haven't heard about before. And the gene names, as you know, are usually very funky. Most of the times they don't really mean something. So this gene was TMEM12 6B. Poor patient, you can imagine, was quite disappointed because there was no diagnosis. And in addition to that, this annoying result of finding two variants of unknown significance in a gene which is a gene of unknown significance. These are the cases where we need new methods to try to figure out what's going on. Because of the uncertainty, and I think based on the questions sent into the Q&A, this happened to several of you listening. The clinicians ordered a muscle and skin biopsy. I kind of wrote it funny, muscle and skin biopsy. When we do an open biopsy, that gives us the chance to collect both skin and muscle samples. You see that the size of the samples doesn't have to be too large. With more modern techniques, we can use really tiny little pieces for functional diagnosis, where we could look into the histology, so we can look also under the microscope for hints about mitochondrial abnormalities. But the most important is what I told you, to measure the mitochondrial proteins, which are responsible for energy production, and we call them the mitochondrial oxidative phosphorylation complexes or respiratory complexes. So in the traditional figure where we depict mitochondria, here you see a beautiful mitochondrial structure. You see this cristae, which are membrane based, and you see here the mitochondrial protein complexes along the cristae. And then when somebody has a muscle biopsy, then these complexes are evaluated by a biochemical laboratory. So when we measure these complexes, we get activity. If you've ever seen a result like that, then it lists all the complexes. It lists like complex 1 activity, complex 2 activity. Sometimes it gives even a measure of 2 complexes working together. And then what you get is a range of activity. And then next to that number, you see the control. So you can see, so the range is for the control, and you see the patient's actual number, and you can tell whether it's within that range or not. So what we saw there, that this patient had a very significantly decreased complex 1 activity, and that's the first complex in the row of proteins making energy in the mitochondria. I enlarged the complex 1 for you to show you that complex 1 is not one protein. It's more than 40 different proteins working together to make energy. So when you look at this name of the gene, what we found, the TMM12-6B, then you can look, okay, so we found the complex 1 activity. Let's look at the genes coding for more than 40 parts of this super complex, but you won't find it. This gene is not one of the genes coding for any of the parts of this super complex, complex 1. So it would have been good if we matched that gene to any of the parts, because then we know, okay, it's malfunctioning, so probably that's why complex 1 is not functioning. But the theory is wrong. Actually, there is no link between the gene we found and this complicated complex 1 structure. I wanted to go back and teach you something else. Most likely when you heard educational talks about the respiratory chain complex, then you learned that complex 1 is the first one, followed by complex 2, and there is like this electron transfer, and then comes complex 3, and then 4, and then 5 will turn around and pump energy. This is not really true. Mitoenzymes are not in a row as they depicted in most of the educational graphs, but they are actually in a bundle. They are the same way how complex 1 is a bundle of 45 subunits all attached together and work together. The complexes are working in a very close proximity as well. They have to find each other, and they have to be physically in contact to work efficiently. So a new method, which has been developed about maybe 8 years ago by a group in Australia, allows us to do complexomics. And complexomics means that you can measure how the mitochondrial complexes find each other, if they're all united, and if they work together or fall apart. So the methodology is quite complicated. You need to have the muscle biopsy homogenized in almost like a liquid form. Then you use a gel that's very similar to how you make a cake and place the pectin on the top of the cake. Just in this case, this gel is used to separate different proteins. You can run them by using electricity, and then depending on the size of the proteins, they separate, and then you can run them to two directions. first one direction, they separate, and then the separated ones, you can run to the other direction. So you will see all the mitochondrial complexes separated, but if they are falling apart, you will see a lot of spots because they want to stay in one complex spot. They will go into little parts. After you did this magic testing, you can use a mass spectrometer. You can cut the gel into pieces and the different parts of the gel, including your muscle biopsies, enzyme, protein particles. You can actually measure how much is measurable out there. So when you get a complexomic results, you get something you probably haven't seen yet on any result you received from your doctor. It's a heat map. It's very complicated, but I will show you how to read such a heat map. If you look at the first column, this is a control. So control is healthy. You see that all the proteins are nicely bundled up in huge super complexes. And you can see complex 1 nicely united. If you look at a patient here, the patient we were talking about, and compare the bundle, you see most of the complex 1 protein is lost. This thin little thing is obviously missing most of the active complex 1, and some parts are completely separate and not even attached to the complex. You see another patient, it's a little different pattern, but in that way similar, that it's missing a lot of complex 1 and it's like scattered and in pieces. And what's surprising that even though the problem is in complex 1, you see that other complexes, for example, complex 4, are also not fully normal. You just compare the visual and you see the abnormality compared to the control patient. So we call this an abnormal complex 1 assembly. In this patient, the proteins in his complex 1 protein didn't find each other, couldn't unite and do the work. That's why we didn't find the gene between any of the genes coding for the complex 1 proteins, because it was not leading to a structural defect, but it led to a so-called assembly defect. But many of the mutations in different mitochondrial genes lead to abnormalities of assembly, or how the proteins are finding each other and working together. And this assembly analysis, or the protein measurements we call proteomics, is a new diagnostic method which we can use to try to functionally prove that in the absence of a functional gene, the consequence is a mitochondrial dysfunction. Is this a proof in itself? No. The doctors in Australia, they also corrected the genetic defect with gene therapy in the dish. And when they gave back the normal gene in the dish to that experimental setting, all the complexes went back to normal, comparable to control. So this is a new way to functionally diagnose mitochondrial disease, mostly used in Australia, in Melbourne, which is a world-famous mitochondrial lab, and starting to be available in a few labs in the United States as well, in addition to complex activity measurement which has still a very important place in mitochondrial diagnostics, especially when the genetic results are normal. Thank you for listening.
Thank you so much, Dr. Marava and Dr. Larson and Dr. Alsharque. What a wonderful evening of talks. And wow, the questions are, amazing questions coming through. Our community really do, a smart group of people out there working and living with this disease, and you really addressed their questions. I loved it. It was wonderful. I'm going to go ahead and pop up our next webcast, where we've unfortunately run out of time for any questions, but I think we've got through a lot of the questions. So I just want to thank our speakers and panelists, and for all the patients and families for attending. And we look forward to you all joining us again on Wednesday, October 23rd. It's 7 o'clock this time. I know we change our times sometimes, so I know that can get confusing, but it'll be 7 o'clock Eastern time, and we will be connecting the dots between common mitochondrial diseases and ultra rare diseases with Dr. Andrea Grootman and Dr. Barantendu Chandra. So we look forward to seeing you all back here next month, and have a great fall, folks, and enjoy and get the word out about mitochondrial disease next week. It is going to begin on Monday, and check out all the activities and get out there and raise awareness, folks. Thank you so very much.